Good evening, everyone. On behalf of the Zoological Association of Burdwan, it is my great pleasure to warmly welcome you to the online summer lecture series for undergraduate studies in zoology. Today, we are honored to have with us Dr. Dipanjan Dutta, an esteemed assistant professor from the postgraduate department of zoology, Hooghly Mohsin College. Dr. Dutta graduated from Burdwan Raj College in 2007 and completed his master's degree with specialization in parasitology and microbiology from the University of Burdwan in 2009. In 2017, he was awarded Doctor of Philosophy from the University of Burdwan for his research on the screening and validation of selected fish gut bacteria as probiotics for Indian major crops. Dr. Dotto has an impressive scholarly record, having published over 10 research papers in highly reputed journals and contributing to 3 book chapters. His achievements include C.B. Jacob Best Paper Presentation Award in 2014 by the Society for Biotechnologists, Junior Scientist of the Year Award in 2018 by the International Academy of Science, Junior Scientist Award at the 5th International Conference on Environment and Ecology held at Christ College, Pune in 2019, and the Emerging Scientist Award in 2023. Furthermore, he has been recognized in the World Scientist and University Ranking 2024 for his significant contribution to the field of microbial diversity in fish gut, the evaluation of gastrointestinal microbiota in fish for screening candidate probiotics, modulation of growth and immunity in fish through the application of probiotic bacteria. With respect and eagerness, I now invite Dr. Dotto to deliver his lecture on digestion and absorption of lipids. Dr. Dotto, the floor is yours.
Thank you, Dr. Ghosh. A very good evening to all the participants. And it is a very nice initiative by the Zoological Association of Burdwan where we are getting this chance to interact with so many students across different colleges. Today, I will discuss on the topic digestion and absorption of lipids. This topic is included in paper CC9, Animal Physiology. In the syllabus, there are 4 macromolecules: carbohydrate, protein, lipids, and nucleic acids. The digestion and absorption we have to study. Out of which, today we will discuss lipids, as I felt that there may be lots of confusion regarding the digestion and absorption process of lipids, as it is a bit different from the other macromolecules. Now I will present my screen. Yes, it's coming. I hope my screen is visible now. Yes. So we are going to discuss about digestion and absorption of lipids. Now, what are lipids? First, a very brief insight about the macromolecule lipids. We all know that lipids are a heterogeneous group of water insoluble organic molecules that can be extracted from tissues by non-polar solvents. By non-polar solvents, we mean such type of solvents which have two different poles or rather dipole. If you want to give some example of non-polar solvents, they are benzene, toluene, chloroform, acetic acid, and others. And we all know that lipids are insoluble in water. That's because water is a polar solvent. Now, the lipids are a major source of energy for our body. We know that 1 gram of lipid gives almost 9 kilocalorie of energy, which is very different from the other two energy producing molecules, that is carbohydrate and protein. Also, lipids provide a hydrophobic barrier that permits partitioning of aqueous contents of cells and subcellular structures. We all know that phospholipids are an integral part of our plasma membrane, and phospholipids makes it possible to demarcate the contents of the cell from that of the extracellular matrix. Fat soluble vitamins A, D, E, and K, they have regulatory or coenzyme role in the body. Prostaglandin and steroid hormones, we all know they play major roles in the maintenance of body's homeostasis. Moreover, lipids also give shape to the body and it protects the internal organs by providing a cushioning effect. So we see there are a diverse function of lipid molecules. Now, when we consume lipids in the form of fat, there are various categories of lipids we all know, like fat, oil, waxes, etc. Out of which, when we consume fat, it is mostly made up of triglycerides. About 95% of fat is composed of triglycerides. The rest are cholesterol, cholesterol esters, phospholipids, and some unesterified fatty acids. These are the composition of the fat we generally consume. Now, the source from which we get lipids are among animals, we get it from meat, especially red meat, butter, ghee, fish, pork, eggs, and among the vegetable sources, we get it from different oils like sunflower oil, mustard oil, groundnut oil, and also from nuts and other things. Now, let's study the background of lipid digestion. The major biochemical process that causes digestion in our body is known as hydrolysis. The term hydrolysis itself implies that it is made up of two parts, hydro and lysis, meaning water and to break down. That means by hydrolysis method, we break down any bond by the addition of water molecules. Now, hydrolysis is exactly opposite to condensation, where two molecules are getting united with the liberation of one water molecule. So, every digestion process of our body is generally included under hydrolysis reaction. Now, when we consider lipids, the chemical bond that is responsible for making up or building the lipid molecules is ester bond. As we have encountered glycosidic bonds in carbohydrate and in proteins, there are also amine bond. Similarly, in case of lipids, it is ester bonds. Now, there are three categories of lipids. The major categories: triacylglycerols or triglycerides, we commonly called, cholesterol ester, and phospholipids. Now we see that triglycerides, triacylglycerol means there is a glycerol backbone in which three carbonyl groups are replaced and forming triacyl. Now the lipases, the enzyme responsible for lipid digestion we found in our body, they are all capable of degrading either in C1 position or in C3 position. No lipase in our body can degrade the triacylglycerol in C2 position. This is quite unique about it. So, the first molecule of triacylglycerol, when there is addition of one molecule of water and one carbonyl group is replaced, we get a free fatty acid from it, and triacylglycerol is degraded to 2,3-diacylglycerol. Now again, when we add a second molecule of water, we again get another fatty acid to be liberated, and 2,3-diacylglycerol now gets converted to 2-monoacylglycerol. Now the process stops here because there will be no further degradation of 2-monoacylglycerol, because as I have said earlier, that at C2 position, no addition of water will be there. So the triacylglycerol or triglycerides will be degraded to 2 molecules of free fatty acid and 1 monoacylglycerol. Now what happens with the cholesterol ester? Cholesterol ester, it is a cholesterol backbone, 27 carbon ring cholesterol backbone, where one ester molecule is just included. And when the specific enzyme for this cholesterol ester hydrolase gets acted, it liberates the free fatty acids from it, and the cholesterol chain gets intact. So cholesterol ester gives one molecule of fatty acid along with cholesterol, the backbone. Now what happens with the phospholipids? The phospholipids, here several categories of lipases act upon phospholipids, and each lipase has its specific bond to be broken. Now in humans we get only phospholipase A2. Now I have pointed out that which bond it is breaking. You can see it, I pointed it by red. Only this bond can be broken in human body by the enzyme phospholipase A2. And when the phospholipids is getting broken down by phospholipase A2, we get another molecule of free fatty acid and the parent molecule, that is known as lysophospholipid. So if we want to recapitulate, when triglycerides are degraded, we get monoacylglycerol or monoglyceride and 2 molecules of free fatty acid. When cholesterol ester is degraded, we get 1 molecule of free fatty acid and cholesterol. And when phospholipid is degraded, we get 1 molecule of free fatty acid and lysophospholipid. So this is the prelude of lipid digestion. Now we'll discuss about the digestion sites. Now in our body there are 3 major digestion sites. One is mouth, second one is stomach, and third is the intestine. Now as we will discuss upon the digestion process, I will also give an insight of the relevant anatomical structures so that it becomes easy for you to understand the mechanism. The first digesting site for lipid is our mouth or buccal cavity. Now you see that our tongue, upon the surface of the tongue, there are several taste buds which are known as papillae. To be exact, there are 4 categories of papillae or taste buds: circumvallate, pungiform, filiform, and foliate. Now, beneath the circumvallate papillae and the foliate papillae, we get a special gland which is exocrine gland, Serra's gland, known as von Ebner's gland. Now, this von Ebner's gland, it is innervated by number 9 cranial nerve, that is glossopharyngeal nerve, and when food comes within the mouth, this von Ebner's gland is stimulated to secrete an enzyme known as lingual lipase. Now, the lingual lipase and next lipase, which we will encounter in a short while, that is the gastric lipase. These two lipases are known as acidic lipases because their pH optimum is in the acidic zone. So, lingual lipase is released from von Ebner's gland. What does lingual lipase do? Lingual lipase is involved in the first phase of lipid digestion. It specially acts upon the short chain triglycerides or the medium chain triglycerides, and it breaks down these triglycerides into its product, that is monoacyl glycerol and fatty acid. Now, this lingual lipase has a very vital role in the digestion process of the newborn babies because the newborn babies consume milk, and milk fat are generally short chain triglycerides and medium chain triglycerides. Now, as the further digestive system of the newborns are not so much developed, a considerable portion of the lipid digestion or milk digestion in newborn occurs within the buccal cavity or mouth itself. In case of adults, however, a small amount of digestion, say 10% of the total fat we have consumed, takes place within the mouth. Just as we are chewing the food, it gets some part of the lipid gets digested within the mouth itself. So as the bolus passes through mouth, we get few triglycerides to be degraded into monoacyl glycerol and fatty acid. The rest, phospholipids and cholesterol ester, they passes as it is. So this is the chemical reaction I have discussed earlier also. Only triglycerides is getting degraded. Now, the next digestion site is stomach. The bolus comes down the esophagus from the buccal cavity and it enters the stomach. Now you see on the right side I have included the picture of a stomach. The stomach has two ends. One part is towards the esophagus and the other where the intestine, small intestine begins. The part where esophagus enters the stomach is known as the cardiac part, and the part where intestine begins from stomach, it is known as pyloric part. Now, the outside of the stomach, we know that it is known as greater curvature. The inside is known as lesser curvature. Now, the fundus portion of the stomach, which is very near to the cardiac part, it contains several gastric pits. What are gastric pits? The epithelial lining of the stomach forms several troughs. You see, the gastric pit, I have included the picture at the bottom. These troughs, they contain three categories of cells. Number one is chief cells, number two is parietal cells, and number three is mucus secreting cells. The parietal cells secrete hydrochloric acid, which maintains the acidic pH of the stomach. And the chief cells, they secrete an enzyme which is very much important for lipid digestion. That enzyme is known as gastric lipase. Now, gastric lipase is again the second category of acidic lipase we find in our body. Gastric lipase is secreted from the chief cells when the chief cells get stimulated by an hormone known as gastrin. This gastrin hormone is released from G cells present in the epithelial lining of the stomach. It is a member of the enteroendocrine system, the G cells. And the G cells, in response to the vagus nerve, they secrete gastrin. And the gastrin causes these chief cells to release the gastric lipase. The gastric lipase requires Ca2 plus as its cofactor for optimum activity. Now, gastric lipase again performs the same function as it has been previously done by the lingual lipase. That is, it degrades triglycerides into two parts, that is monoacyl glycerol and fatty acids. Now, so the bolus, when it will get out from stomach through the pyloric end or the pyloric sphincter, so again a portion of the total triglyceride we have consumed is getting degraded. The cholesterol ester and phospholipids are staying as it is. So this is the again the same chemical reaction is occurring here also, as we have found within mouth. Now we come to the next digestion site, that is the small intestine. Now let's study some anatomy of the pancreas. You see that our liver, it is made up of two lobes, the right hepatic lobe and the left hepatic lobe. A duct comes out from each lobe, which is similarly named right hepatic duct and left hepatic duct. Now in this diagram, you will not find the picture of liver, but you see that right and left hepatic duct is mentioned here. The right and left hepatic duct unites to form a common hepatic duct. Now, there is another organ known as gallbladder. This gallbladder has a duct which is known as cystic duct. Now, the cystic duct opens with the common hepatic duct to form the common bile duct. Now, this common bile duct will come straight into the duodenum and will open into a trough-like structure, just a depression-like structure. It is known as ampulla, hepatopancreatic ampulla or ampulla of Vater. On the other hand, the pancreas has a major pancreatic duct which is known as duct of Wirsung. Now, this duct of Wirsung, it joins the earlier duct, that is the common bile duct, again into the ampulla of Vater. Now, in few persons, you will find another accessory pancreatic duct which is known as duct of Satorini. But this accessory pancreatic duct is not present in every individual. Wherever it is present, it opens into another accessory ampulla, not into the ampulla of Vater. Now, in the ampulla of Vater, where these two ducts gets emptied, that is the common bile duct and the main pancreatic duct, there is a sphincter of smooth muscles. This sphincter is known as sphincter of Oddi. Now, what happens when food comes within the, or rather bolus comes within the proximal part of the intestine, then another enteroendocrine cell, as we have encountered G cell in stomach, now it's turned for I cell of duodenum. The I cell is again an enteroendocrine cell, and the I cell secretes cholecystokinin, CCK. Now, the primary function of cholecystokinin is just to open the sphincter of Oddi. Now, as the sphincter of Oddi is opened, the content of the common hepatic duct and the duct of Wirsung will be discharged into the lumen of the duodenum. So, this is the anatomy. Now, what are the secretions of pancreas in relevance to lipid digestion? Or to be very exact, these are the secretions of the exocrine part of the pancreas, which are known as acinar cells. There are four things included within the pancreatic juice: pancreatic lipase, phospholipase A2, cholesterol esterase, and procolipase. On the other hand, the gallbladder secretes bile. The bile is composed of two things. One is lecithin, the other is bile salt. Now, lecithin is a type of phospholipid. So, all these molecules, all these secretions together causes the final digestion of lipid. Now, the biggest problem of lipid digestion is that you just consider the situation that within the lumen of the duodenum, the chyme is floating. The chyme is a watery secretion, and within that chyme, large fat globules which contains triglycerides, which contain cholesterol ester, which contains phospholipids, along with monoacylglycerol and fatty acids. Now, these are largely hydrophobic molecules. Now, as these are hydrophobic molecules, they tend to aggregate together and form a globule. Now, what is the problem? There is large globules are floating on the lipid layer, on the intestinal lumen. And unfortunately, the lipases, pancreatic lipase, which is an alkaline lipase, this is hydrophilic in nature. It is within the watery chyme layer. And there is very little lipid to water interacting layer is very less. So, this lipase unfortunately cannot act upon the fat globules because it is getting very little space to act. So, if we want to digest the lipid molecules, we have to make sure that it sinks within the chyme so that our lipase gets enough space to act upon this. This method by which we will be able to make this fat globule sink within the watery secretion of the intestinal chyme is known as emulsification. Now, what are the key players responsible for emulsifying? Very important is bile salts. As I have said earlier, the bile is composed of two components. Number one is lecithin and number two is bile salts. Now, the bile salts, they are derivatives of cholesterol. Very simple. They are again derivatives of cholesterol. Here, there are two amino acids, glycine and taurine, which will act with the cholesterol ring by amide linkage to form two categories of bile salts. See, the cholesterol, first it acts upon by an enzyme known as 7-alpha hydroxylase. It forms the first side chain, that is 7-hydroxycholesterol. This 7-hydroxycholesterol gets converted into two categories. One is cholic acid, the other is chenodeoxycholic acid. There are a big process involved within this, but this is not the purview of the present discussion. You can see that there are classical pathways, there are alternative pathways for generation of these bile acids from cholesterol. Now, the cholic acids, when glycine and taurine reacts with cholic acid, we get two components, that is glycocholic acid and taurocholic acid. And when they react with the chenodeoxycholic acid, we get another two component, that is glycochenodeoxycholic acid and taurochenodeoxycholic acid. All these are bile salts and known as primary bile salts. On the other hand, these primary bile salts are sometimes acted upon by different gut microbiota to generate secondary bile acids from this. The secondary bile acids are deoxycholic acid and lithocholic acid. Now, in humans, the ratio of the glycine to taurine conjugates is 3:1. That means we find that glycine derivatives are large in number in comparison to taurine derivatives. Now, I have tried to explain this by diagrams. You see that our intestinal chyme is watery, and the fat globule composed of largely three molecules: triacylglycerol, cholesterol ester, and phospholipids. Large fat globules are floating upon this. Unfortunately, pancreatic lipase, which is hydrophilic, it is present within the intestinal chyme and it cannot act on the fat globule. Now, the first thing, as the process of emulsification triggers, first molecule that triggers the emulsification process is lecithin, another component of bile. Now, lecithin is a very good molecule because it is amphipathic in nature. You see that what are amphipathic molecules? That means one end of this is hydrophobic, the other part is hydrophilic. Now, this lecithin can sit upon the fat globule because hydrophobic, there are same interactions, and the outside is hydrophilic. Now, as the outer layer gets converted from hydrophobic to hydrophilic, now the fat globule starts sinking within the intestinal chyme, because this time it will not repel the water, but it will get attracted by water. The second thing that comes into play are the bile salts. These bile salts does nothing but sits on the lap of lecithin, thereby further increasing the molecular weight of the whole component. So thereby it gets further sink to the bottom, this fat droplet. Now, again, the pancreatic acinar cells, the fourth component of the pancreatic juice, that is procholipase, is released from the pancreatic acinar cells. Now, this procholipase is a zymogen. It is an inactive form of an enzyme. And after it being secreted, this procholipase is converted into its active form, that is scolipase, by cleaving the side chain by another enzyme which is instrumental in protein digestion, that is trypsin. Trypsin, what it does, it just cleaves the side chain of procholipase to generate the active form of the procholipase known as colipase. Now, the colipase is a coenzyme for pancreatic lipase, and as soon as colipase now sits upon this total molecule, now it becomes very easy for pancreatic lipase to exert its effect. So, what will pancreatic lipase do? The pancreatic lipase will degrade every molecule of triglycerides, the remaining molecule, of course, which have escaped earlier digestion by lingual lipase and gastric lipase. Now, this time it will be degraded, will get lots of triglycerides and free fatty acids. Cholesterol ester hydrolase released from pancreas will degrade the cholesterol ester to liberate fatty acids and cholesterol, and phospholipase A2, another of the member of the pancreatic juice, it will degrade the phospholipids to generate the fatty acids and lysophospholipid. Now, as the digestion is going on, the center core is getting smaller and smaller because lots of compounds are getting degraded. So earlier, the first molecule was fat globule, which was the largest in size. Then as soon as the digestion starts, it decreases in size, it gets shortened from fat globule to fat droplet, and now it gets further decreased in size to form micelle. Now the center core is micelle, where we find phospholipids, we find cholesterol, we find free fatty acid, we find monoacylglycerol, out of which lecithin is sitting. Above lecithin, bile salts are sitting. And now another good boy comes and sit upon this. They are the fat soluble vitamins A, D, E, K. They need to be transported. So as they are fat soluble, they are also getting problem in the intestinal chyme. So they sits upon this, and together they forms the micelle. Now this micelle now enters the enterocytes. What are enterocytes? Until this, the whole process was continuing within the lumen of the duodenum. Now, the epithelial lining of the duodenum, there are simple columnar cells with apical microvilli forming brush border. These brush border cells are known as enterocytes. Brush borders are nothing but cytoplasmic extensions. Now, these enterocytes will engulf these micelles. And one thing they will discard, that is the bile salts. The bile salts component gets straight discarded, and they enters the enterohepatic circulation, and they go straight to liver. They will again be recycled in the earlier process. So the bile salt goes out. We are left with phospholipids, cholesterol, fat soluble vitamins, free fatty acid, and monoacylglycerol. And this whole thing, the micelle enters the cytoplasm of the enterocyte. Now from the cytoplasm, the micelle goes to the smooth endoplasmic reticulum of enterocytes. Now smooth endoplasmic reticulum, what it does, it again converts the free fatty acid and monoacylglycerol into triglycerides. And cholesterol gets again esterified to form cholesterol ester. Until now, which the breakdown products are again reunited to form the earlier products. This is quite interesting. Now, we get again phospholipids, cholesterol, cholesterol ester, triglycerides, and fat soluble vitamins. Now, this thing enters the rough endoplasmic reticulum. From the smooth endoplasmic reticulum, the whole thing goes to rough endoplasmic reticulum of the enterocyte. Now, the rough endoplasmic reticulum keeps a protein coat to it. And another thing, a lipoprotein, which is known as ApoB48, apolipoprotein B48. These two things they keep. And this whole thing will now be called as chylomicron. Earlier it was known as micelle. Now it will be known as chylomicron. There are now this chylomicron will enter from rough endoplasmic reticulum to the Golgi complex. Now we all know that Golgi is very expert in packaging. So the Golgi will further package this chylomicron, and it will again give another, there will be another accessory lipoprotein like ApoC, like ApoA, like ApoE, which are accessory lipoproteins to ApoB48. Now this chylomicron, the whole packaged chylomicron, which is very good looking, complete, it has been completely packaged by Golgi. Now it gets exocytosed from the enterocytes. Now you see that enterocytes, they have two end. One is towards the lumen, the other is towards the extracellular matrix outside of the intestine. So from the end, which was towards the lumen, they have taken the micelle, and after all these things, all this packaging, all this synthesis, now they are driving out the chylomicron from the other door. The enterocytes are exocytosing the chylomicron. Now what happens when the chylomicron comes outside the exocytosis? In case of carbohydrate and protein digestion, it was taken up by the blood capillaries. But unfortunately, the size of the chylomicron is so large that blood capillaries will be not able to take up the chylomicron. The chylomicron size is nearly more than 1 micrometer, you see. So so large a molecule cannot be taken within the blood capillaries. So what we will do? The chylomicrons will then be taken by another type of capillaries which are known as lacteals. What are lacteals? Lacteals are lymphatic capillaries. They are earmarked for, they are dedicated for lipid transportation. Now these lacteals take up the chylomicron. Now all these lacteals, there are numerous lacteals like the blood capillaries, and all these lacteals which have taken up the chylomicron, they are emptying it into the main lymphatic duct, which is known as thoracic duct. Thoracic duct is the main lymphatic duct. Now thoracic duct will carry this chylomicron. And now the problem will not be solved if the chylomicron stays within the lymphatic capillaries itself, because unless and until it comes in within the blood, it will not reach every cell, and so we cannot be able to produce energy from this. So we need to transfer this chylomicron from lymphatic capillaries to blood capillaries. So this thoracic duct, or the main lymphatic canal, it comes into a junction where the left internal jugular vein and the left subclavian vein unite to form the left brachiocephalic vein. The region is near our shoulder, where the internal jugular and the subclavian are uniting to form brachiocephalic. Now at this triad junction, the thoracic duct is emptying its contents. So the chylomicron comes now to the brachiocephalic vein, and from the brachiocephalic vein, the chylomicron will have two destinations. One will be the skeletal muscle and the other will be the adipose tissue. When it will be transporting towards either these two junctions or these two destinations, the wall of the capillary endothelial layer, there is again an enzyme present on the inner wall of the capillary endothelium, which is known as lipoprotein lipase. Now this lipoprotein lipase will again degrade the triglycerides of chylomicron into free fatty acids. Now these free fatty acids, when it will reach the adipose tissue, it will be again resynthesized into triglycerides utilizing the glycerol which we obtain from glycolytic pathway. It will be tagged with the glycerol to form triglycerides, and this will be the fat storage of our body. When we were starving, our body will provide energy from these triglycerides stored. And when the free fatty acids will reach the skeletal muscle, we will generate ATP from this. Now the triglycerides and the free fatty acids, they are gone from the chylomicrons, and the remainder which are left to say cholesterol, cholesterol ester, and lysophospholipid and others, These are now known as chylomicron remnants. These chylomicron remnants will finally reach liver and they will convert it into VLDL or very low density lipoprotein, which is the final end product of lipid digestion. I hope that I have been able to give an insight the whole process, and I will be very happy if you ask few questions where there is doubt regarding this. So any question is welcome from the participants.
Dear students, if you have any question.
I think they don't have any question.
Okay. We are deeply grateful to you, sir, for accepting our invitation and sharing your valuable knowledge with us. Dr. Dotto has expressively discussed the digestion and absorption of lipids. I extend my heartfelt congratulations to you for delivering such an informative lecture, which I am confident will greatly benefit of our students in their academic studies. We also extend our thanks to the students for their active participation. With this, we conclude today's lecture session. Thank you, everyone.
Thank you. Thanks a lot.